complex process goes anaerobically, which splits sugars up in cells to make ATP. With lysis to mean breaking, and glyco to be sugar, we refer to glycolysis in your body. Overall, glycolysis consumes 1 glucose. The final product yields 2 times a triose. Called pyruvate, but the net's more grandiose, 2 ATP and NADH from 1 dose. This occurs at the expense of 2 NAD+. Without their daring, we'd lack ATP in us. Now sit back and relax, rather like a rebus. Enjoy the artistry, which now we'll discuss. The prima ballerina, glucose is 6-membered. Like 2 trios in 1 ring, their edges still blurred. Low energy, it's chill, soon to be dismembered. In the cytoplasm, first a P group is transferred. Hexokinase is the one that gets this job done by cleaving ATP to add a phosphate 1. Now glucose 6-phosphate, but this enzyme has fun and can phosphorylate fructose, which may stun. Back to G6P, the ring is made smaller sized. From a pyranose to a pentose, it's catalyzed by phosphoglucose isomerized, specialized, to form fructose, with C6 still phosphorylized. This fructose 6-phosphate, that's F6P to U, gets a pickup by phosphofructokinase 2, a higher energy state. As F6P grew, this consumes ATP. Now the P counts at 2. Here we arrive at fructose 1,6-bisphosphate. It's the largest we see, the greatest of the great. It ends not here, for 2 trios halves is its fate, and hence part 3 carbon soulmates now separate. Aldolase is the one that cleaves these bonds apart into 2 different molecules, both works of art. The cell does not use DHAP, which is not so smart. However, the GAP now has a new start. Dihydroxyacetone phosphate envies her brother, for GAP is much preferred by another. So triose phosphate isomerase will smother the ketose DHAP to switch to the other. Now DHAP runs the same path as its friend, for the triose twins take the same course as we wend, through glycolysis' second phase on which we depend, for ATP and hypoxia for us then to spend. Glyceraldehyde 3-phosphate dehydrogenase, GAPDH, consumes NAD+ always. Using inorganic phosphate, it then conveys the P group to the triose, its energy state raised. GAP becomes 1,3-bisphosphoglycerate. Due to the GAPDH, there's a second phosphate. We've consumed a total of 2 ATP at this state, but soon we reap back 4 from the glucose we ate. Our first payback occurs as we form 3PG. As we know, phosphoglycerate kinase is key to breaking even with our count on ATP, but we must change before we see more charity. Our good friend phosphoglycerate mutase is cued in, who conducts a quick change circus to much chagrin. Switching the phosphate from the 3rd carbon wherein, it moves to the 2C and is now held within. There's 2PG, triumphantly the enolase shout. We knew you'd make it in good time, without a doubt. And while they waited, they became a thirsty lout. So 2-phosphoglycerate drops water before it sails out. Cast off by enolase, we've arrived near the end, to the poor soul pyruvate kinase, who's tasked to wend off a P group from their phosphoenolpyruvate friend. The net of 2 ATP from glucose is the trend. At the finale of the glycolysis pathway drama lies a new beginning for more molecule trauma. Here groups pyruvate, whose fermentation's mama. And as the story goes on, will end here with a comma. Cells need glycolysis, but the story goes on. Through the TCA cycle and electron, transport chain, oxidative phosphorylation, for metabolized glucose to be truly gone. While complete glucose oxidation yields 38, while glycolysis yields but 2 ATP, not so great. But quick cancer growing needs energy on its plate, efficient glucose use, and fastest ATP making rate. Cancer prefers glycolysis. How is this so? Shouldn't they have the most ATP on the go? Cancerous tumors grow quickly, this we surely know. How can 2 ATP be enough for it to grow? This initially counterintuitive observation is known as the Warburg Effect, and upon this topic we focus our final project on. How is glycolysis different in cancer cell spawn? We mentioned the yield of 19 times less ATP. We also know glycolysis runs anaerobically. This fact and the cancer tumor hypoxia agree. The cancer must upregulate something to make energy. Ostis et al., back at the 2000-year tick, a J-Bio Chem paper was their top pick. They studied how higher expression of c-Myc leads to deregulation of most things glycolytic. c-Myc is an oncogene turned up in a cancer core. It shares a consensus sequence with the protein called CHOR, a carbohydrate response element which opens the door to transcribe glycolytic enzymes yet even more. Mice livers were injected with an adenovirus shot. For LAC-Z control or c-Myc expression they sought. The hepatocytes were then collected for Western blot, where Ostis then compared the glycolytic lot. In Part B of Ostis Figure 1, here we now cite, shows the expression of c-Myc in the murine hepatocyte. Very little expression on day 3 is something to cite, but substantially more each day up to day 5, they write. In Part C of the same figure, we see 6 enzymes, the first of which is GLUT1, which is needed oftentimes for glucose uptake into the cell, for which it primes, glycolysis, and with c-Myc its expression slightly climbs. PFK, which you'll remember, adds a phosphate to F6P so it can form its FBP mate, and from day 3 to day 5 it will upregulate when c-Myc is around, which is the cancer state. We remember GAPDH, which will change GAP to form 1,3-BPG and uses NAD+ in exchange. GPI forms a pentose, causing hexose to rearrange, and both are upregulated with c-Myc does arrange. PGM shifts a P group from 3-carbon to 2, while enolase takes water from 3PG2, form PEP, and without further ado, both are upregulated by c-Myc in the cell too. You may have noticed that this only occurred on the right side with c-Myc adenovirus injection. The left side showed LAC-Z negative control expression and basal levels of each enzyme's translation. To conclude Ostis Figure 1, Part B and C, upregulation occurs for the majority of glycolytic enzymes. Modest GLUT1 specifically is later shown to have even higher activity. Pyruvate kinase of the glycolytic genes is last in line, so if PK activity is low, it means for the upstream intermediates, it's no small beans. They'll back up due to the other enzyme machines. There are two isoforms of this enzyme, M1 and M2. Adult cells use the more active M1, which we construe to mean pyruvate goes on to be further oxidized too. Remarkably, cancer cells use PKM2 to make do. PKM2 is the embryonic isoform due to splicing events. Later we'll see it's regulated by FBP's presence. It also acts as a nuclear transcription factor and contends itself to upregulate several genes in different segments. For it activates beta-catenin, which is independent of Wnt, as well as interacting with HIF1, playing an essential stint in the cellular response to hypoxia, which gives you a hint of how important a role PKM2 plays. This is not a misprint. Figure 1 from Christophe et al., published in Nature, is a Western blot with proteins treated to denature. Tumors from 4 mice were collected, and to be sure, they included a control of normal mammary tissue pure. Another control to ensure even protein per load, beta-actin was detected, shown evenly here below. The control showed the ample PKM1, although, in tumors, M2 was more prevalent, we now know. Over the tumor progression in mammary tissue, PKM2 protein expression increases in vivo, and though this experiment they've demonstrated a breakthrough, there is alternate splicing occurring between the two. Christophe demonstrated this distinction in her second design, lung carcinomas A549 and H1299, while 293T is a transformed embryonic kidney line. Fourth, the HeLa cell is a cervical carcinoma that grows just fine. Additionally, MCF10A is an immortalized cell taken from a source of breast epithelial. They use muscle as the normal sample as well as anti-GAPDH, which is even across the gel. Here we see the tumor lines express PKM2, all 5 of the ones we described above to you. Only in the muscle, where it is normal too, express PKM1. It does, and the results ensue. We continue with Christophe's work in Figure 3, which is divided into parts A, B, C, and D. The cell lines are both the lung types used previously, as well as MCF7 and SN12C. Part A examined whether a lung tumor cell grown in hypoxia or glucose, but the second is not shown. In the condition of low oxygen, they asked, did its own doubling rate change by M1 or M2 rescue alone? The doubling rate is a marker of proliferation. A faster rate is shown by a shorter duration. They compared their M2 knockout tumor creation after rescuing the cells with PKM2 or M1. The lung cancer cell rescued with M1 isoform had a longer doubling time than was the norm. For M2 in both of the treatments that she did perform, the control showed that untreated cells conform. Oligomycin was used, which is a specific inhibitor of mitochondria that they stick in the media to prevent ATP synthase quick, and a significant difference was caused by this trick. Part B and C use all 4 cancer cell lines. We've discussed here above, although it's important to define, that MCF7 is not an invasive, but the rest are malign. They used an oxygen and lactate testing design. Like in A, they treated the cells with M1 or M2 to recover, although the scales are different from one to another. We see a trend when comparing M1 to its M2 brother. It used more O2 and made less lactate than the other. They found a statistical difference that they could believe in, and they found the rate of oxygen consumption is even. and the non-proliferating cancer cell line, MCF7. These clean results could cause a scientist's heart to leaven. Part D compared metabolite levels lastly. They were determined by liquid chromatography, which was followed in series by mass spectrometry. Those to note were lactate, pyruvate, and FBP. By FBP, they mean fructose 1,6-bisphosphate, which also included fructose 2,6-bisphosphate. They are indistinguishable, eluting at the same rate, as well as having similar structure and weight. There were others tested that didn't change like planned. These include citrate, isocitrate, fumarate, and oxaloacetate and malate, whose changes were not grand. We know these are part of the TCA cycle offhand. To conclude Christoph's finding, something quite very interesting happened with FBP in every rescue attempted with M2, wouldn't you agree? In cancer, FBP may regulate PKM2 activity. PKM2 is linked to the Warburg Effect, but there's still work to do, Christoph checked. There are multiple routes M2 may work, they reflect, where M2 and glycolysis do connect. But the take-home message is not one to forget. A switch in metabolism is what M2 will get. Aerobic glycolysis is a cancer preset. We see this preference only in tumors as of yet. The splicing pattern is how PKM1 and M2 differ. Exon 9 and 10 are mutually exclusive, they concur. PKM1 keeps E9, and E10 does M2 prefer. How does this preference arise and occur? The first two papers showed that c-Myc is the norm in cancer and upregulates glycolysis to perform. We also know that PK is found as the M2 isoform. We now discuss how c-Myc regulates this PK form. With a publication in Nature in 2010, David et al. Said, PKM2 is up in cancer, but what then? Is it regulated by, and how does this splicing happen? They found 3 new proteins as well as c-Myc again. The authors used UV cross-linking light to identify proteins binding the exon 9 splice site. With the purpose of inhibiting E9 inclusion, they write, they identified these proteins, which we now cite. 2 main heterogeneous nucleoribo nucleoproteins, HNRNP for show, and a polypyrimidine tract binding protein, although we'll call it PTB, so to the results we now go. HNRNPA1 and 2 bind E9's 5' splice location, while HNRNP1 or PTB took the boundary of this exon. They covered up sites that are critical to exon 9 inclusion, and therefore is left out and E10 is tacked on. c-Myc, the oncogenic transcription factor, increases the expression of the 3 HNRNPs more. The direct role of c-Myc, the authors wanted to explore in HRNP expression and splicing of PK. So in figure 4, the NIH3T3 fibroblast cells were transfected with control snRNA, or that which was c-Myc targeted. Short hairpin RNA that would make the cell c-Myc depleted, while the control shows basal levels that are not affected. This is part A, where we see that the c-Myc loss decreased protein levels of HNRNPs like a boss. However, there is no change in the splicing factors across, those not linked with PK, so here they are dross. Those unaffected were called ASFSF2, as well as CPSF73 and through. Both test conditions showed similar expression too. Last but not least, PTB was affected in their view. The previous was immunoblotting. Now in part B, David et al. Looked at mRNA by RT, PCR, which requires reverse transcription as he, then compared the same 2 conditions as previously. beta-2 microglobulin, the control, B2M if you please, mRNA levels of PTB and 2 hNRPs were also quite low with c-Myc knockdown, and these suggested that it regulates the transcription of their genes. In Part C, they compared the mRNA ratio of M1 to M2 in the conditions we know. More PKM1 is there when c-Myc is knocked low, while PKM2 reigns supreme when cancer cells grow. Last but not least, Part D is a model showing PK splicing regulation in the cell. By PTB, hNRPA1 and 2, they compel us to see they define the intron regions quite well. Abbreviated as U1 and U2, we see adult tissues show low expression of all three. This allows recognition of exon 9 by machinery that splices, and now exon 10 is the absentee. In proliferating cells, cancer and embryonic, upregulated PTB A1 and 2 bind intronic, splicing signals flanking E9 and causing chronic exclusion. Therefore, E10 is included. How ironic. To the authors, this makes sense in their conclusion. They suggest that E10, an alternatively spliced exon, is poorly recognized without intron definition, which is provided by the proteins in this discussion. Lastly, the fact that depleted of the protein leads to the switching of PKM1 must mean the RNA binding protein's control is split between repressing E9 while activating E10, as seen. During this project, we had so much fun describing how glycolysis is usually run. Then we tied in c-Myc that regulates expression, so the tight regulation of glycolysis is undone. We now know this is called the Warburg Effect, where cancer uses aerobic glycolysis unchecked. In times of hypoxia, this will protect the cancer tumor cells just like we'd expect. We know intermediates in glycolysis are upregulated in cancer, but we can't dismiss that they are also precursors in anabolic processes, which also include protein and lipid synthesis. A buildup of the intermediate FBP was key to regulate the pyruvate kinase, we agree. In growing cancer, PKM2 was shown to be, and they doubled faster than normal cells would be. In conclusion, glycolysis enzymes are higher, and all three known proteins get M2 splicing to occur, all of which are controlled by c-Myc in cancer. These articles are from JBC, Science, and Nature.